Hello everyone, welcome to the channel Microbial World. In this video, we will be discussing about gram staining. So before we look into the topic, we need to understand about staining. Staining is a technique used in microscopy to enhance contrast in the microscopic image. Stains and dyes are frequently used in biological tissues for viewing, often with the aid of different microscopes. A stain is a substance that adheres to a cell, giving the cell color. The presence of color gives the cell significant contrast, so they are much more visible. Different stains have different affinities for different organisms or different parts of organisms. Bacteria have nearly the same refractive index as water. Therefore, when they are observed under a microscope, they are opaque or nearly invisible to the naked eye. Different types of staining methods are used to make the cells and their internal structures more visible under the light microscope. Stains are classified as simple stain, differential stain, structural or special stain. Gram staining. Gram staining is a differential staining method. It is used to identify and differentiate bacteria into two groups, that is, gram-positive and gram-negative bacteria. This classification is based on the chemical composition of the bacterial cell wall. Gram staining procedure was developed in 1884 by Hans Christian Gram. Reagents used in gram staining. The gram stain uses four different reagents. Primary stain crystal violet. This violet stain is used first and it stains all the cells purple. Mordant grams iodine. This reagent serves not only as a killing agent but also as a mordant, a substance that increases the cell's affinity for a stain. The reagent does this by binding to the primary stain, that is crystal violet, thus forming an insoluble complex. The resultant crystal violet iodine complex serves to intensify the color of the stain. At this point, all the cells will appear purple black. Decolorizing agent, ethyl alcohol 95%. This reagent serves a dual function as a protein dehydrating agent and as a lipid solvent. So in gram-positive bacteria, it acts as a dehydrating agent, and in gram-negative bacteria, it acts as a lipid solvent. Its action is determined by two factors, the concentration of lipid and the thickness of the peptidoglycan layer in the bacterial cell wall. Counterstain safranin. This is the final reagent that is used, and it stains those cells that have been previously decolorized pink in color. Since only gram-negative cells undergo decolorization, they may now absorb the counterstain. Procedure of gram staining. Take a clean grease-free slide. Prepare the smear of suspension on the clean slide with a loopful of sample. Air dry and heat fix. Crystal violet was poured and kept for about 30 seconds to 1 minute and rinse with water. Flip the grams iodine for 1 minute and wash with water. Then wash with 95% alcohol or acetone for about 10 to 20 seconds and rinse with water. Add safranin for about 1 minute and wash with water. Air dry, blot dry and observe under microscope. Principle of gram staining. The cell wall of gram-positive bacteria have a thick layer of peptidoglycan and the lipid content is low. As we said before, the decolorizer acts as a dehydrating agent in gram-positive bacteria. So decolorizing the cell causes this thick cell wall to dehydrate and shrink, which closes the pores in the cell wall. So the permeability of the cell wall decreases and this prevents the stain from exiting the cell. So the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram-positive bacteria. As a result, gram-positive bacteria appears blue or purple in color. Gram-negative bacterial cell wall also takes up the crystal violet iodine complex. But due to the thin layer of peptidoglycan and thick outer layer which is formed of lipids, the crystal violet iodine complex gets washed off. As we know, the decolorizer acts as a lipid solvent in gram-negative bacteria. When they are exposed to alcohol, decolorizer dissolves the lipids in the cell wall. This increases the permeability of the cell wall, which allows the crystal violet iodine complex to leach out of the cell. Then when again stained with the counterstain safranin, they take the color of the stain and appear red in color. So how can we interpret the result? Gram-positive bacteria appear blue or purple in color and gram-negative bacteria appear red in color. Examples of gram-positive bacteria include Actinomyces, Bacillus, Clostridium, Cornybacterium, Enterococcus, Gardnerella, Lactobacillus, Listeria, Mycoplasma, Nocardia, Staphylococcus, Streptococcus, Streptomyces, etc. Gram-negative bacteria include Escherichia coli, Salmonella, Shigella, and other Enterobacteriaceae, Pseudomonas, Moraxella, Helicobacter, Stenotrophomonas, Dellovibrio, acetic acid bacteria, Legionella, etc. So this is all about gram staining. Thank you.